Hello and welcome. My name is Dana Dayton, and I am the Director of Communications and Engagement at the Melanoma Research Alliance, and I will be moderating this webinar. We'd like to thank all of you for joining this MRA meetup called CAR T-Cell Therapy, another in a series of webinars designed to engage, educate, and most importantly, empower patients and caregivers. Before we get started, I have a few housekeeping things. By joining this webinar, you understand that this will be a public event and recorded as a resource for others that will live on the MRA website. Portions may also be shared via MRA social media channels. The content should not be used as a substitute for medical advice, diagnosis, or treatment. Please consult your healthcare provider about any questions you may have regarding a medical condition. It is your choice to be on or off camera. Please keep your line muted and other devices silenced should you come off mute to ask a question. And please be respectful of the experiences, opinions, and questions of others. If you would like to ask a question, please use the raised hand feature on Zoom, and we will do our best to call on you. So now, without further ado, it is my pleasure to welcome to our panel Dr. Anusha Kalbasi, oncologist and researcher at Stanford University. Dr. Kalbasi, before we get started, do you want to say a few words about yourself?
Happy to. Thanks so much, first of all, for having me. I'm really looking forward to the discussion today. I'm a oncologist, as you mentioned, specifically a radiation oncologist, but my research has been focused largely on how cancer and the immune system interact and how we might be able to use the immune system to fight cancer. And as part of that, we use T cells that are either grown from patients or engineered from patients to do that. And that's what we'll be talking about today.
Great. Well, this is such an exciting area of science. One more thing before we get started, I wanted to thank our MRA team, Renee Orsioni, Digital Engagement and Communications Manager, and Yasmin Idrisi, who is our digital media intern, for helping to set this up, helping to promote it, helping to do the registration. And later on, you will see some tidbits that are posted socially. And of course, the recording will be on our website. So let's get started. Dr. Kalbasi, the first question that I have for you is more general. Before we dive deeper into CAR T and the Stanford trial, can you please discuss clinical trials for a bit? What role do they play in a melanoma, and why are they so important?
Yeah, absolutely. I think clinical trials are kind of the bedrock of our ability to make advances in medicine. And so, we think of discoveries or ideas in the laboratory to come up with new drugs or new approaches to treat patients, whether it's patients with cancer or patients with other diseases. But in order to then develop those drugs, that's one big challenge. But once we have drugs or approaches that are ready to go to patients, we have to be able to test whether those drugs are safe, whether they're effective, and whether they're even more effective than drugs that are already out there or approaches that are already out there. And so, the FDA is the big body that helps us, regulate whether drugs that are administered to patients are safe, are effective. And so we generate data from clinical trials in order to be able to prove to the FDA that drugs are safe and drugs are effective and more effective than standard of care treatments. So it's a process. It takes a long time to go from idea to getting a drug to patients in a way that is approved. But it's an important process because it's what keeps us from having drugs out there that aren't effective or dangerous to patients. It makes sure that we have a high bar for what we give to our patients. At the same time, clinical trials offer an opportunity, both for patients and for the community, to learn about a disease, to learn about how diseases respond to different treatments. Also gives patients an opportunity to try promising new treatments that may not be available for several years. So while it serves as this kind of pipeline to develop drugs that are approved, along that pipeline, it's an opportunity. It's an opportunity for patients to get access to exciting new treatments, but it's also an opportunity for researchers to learn about the diseases that we're treating and such. And there's generally like a kind of a stepwise process in which these trials are done. And I can talk a little bit about that.
Great. That's really helpful, and we'll get to those phases in a minute because I think it's important for people to understand them. Maybe before we get there, though, you can talk a little bit about what questions patients should go to their physicians with around clinical trials. I think there's a perception that sometimes they're only used for like kind of last ditch efforts, but that's not always the case. And if they're interested in learning more, how would they find out about them? What do they need to know? Um, General things like that.
It's a great thing to be plugged into clinical trials from an early time, especially for patients with cancer, because nowadays trials can happen at any stage of the game, right? At any stage of the disease process, when patients are first diagnosed, when patients are first go through their first treatment and maybe treatment doesn't work as well as patients who've gone through several treatments. So depending on where you are in the disease process, the trials may be available for a particular patient. Questions to ask are kind of, what is the standard treatment? I think that's really important to know. Okay, what are the standard treatment options, and what is the success rate of the standard treatment option? Because if the standard treatment option is very successful or has a high success rate, then maybe somebody's willingness to do a clinical trial may be lower. But sometimes standard treatment options, even though they're standard, are not all that effective. And so understanding, what is being offered to you or your family member, and what is the chance that will be successful is critical. And then once you know that, and then understanding what is the landscape of treatment options out there, and there are more and more every day, and they encompass different categories of treatments, from immunotherapy to targeted therapy to cell therapy. So kind of getting a general idea of what is entailed with each in each of these, and what is the commitment to being involved in a trial. So what's available, and then what's the commitment, and then what is the excitement around that? And that you really have to rely on your doctors, and sometimes you'll have to consult with multiple doctors, because not every physician is in tune with all the trials that are going on at any particular time. So it does take a long time. It's not something that you can tackle in one visit to get a sense of what's going on. That's why I think it's really important from early on for patients to start to educate themselves about their disease process. What are the clinical trials and treatments that are being tested, so that when the time comes for actually considering a clinical trial, you're not, starting from basics. At that point, then you kind of have an idea of what to expect, what are some basic options. There are a lot of resources online to look for trials, clinicaltrials.gov, other things. But word of mouth, organizations like MRA and disease focus groups. So these are all great places to kind of get a sense of what's going on.
Yeah, thank you for mentioning that. Here at MRA, we have our own clinical trials finder on our website, which is a great resource for patients. If they want to enter some specific details, it'll help crank out things that might be applicable to them. One thing that's important, I think, to any patient with any indication is to understand where they are in their journey and where the trials are in their journey. There are specific phases to a clinical trial, right? Can you talk to us a little bit about those and how they differ and what that means for patients?
Yeah, and I think we have a visual to kind of capture this that we can pull up at the same time. But generally speaking, I like to divide it into three groups: trials that are done early on to just test primarily whether a drug, for example, if we're talking about drugs, because not all trials are related to drugs. Let's just be clear about that. But if a trial is related to a drug, then the initial phase, or phase one, is just to test whether a drug can be safe, and whether the drug does what it's supposed to do in terms of where it goes in the body and how long it lasts, how long it takes before it's excreted out of the body. Some basics, just learning about how the drug works. Now, in that phase one, we're obviously also hoping that the drug will be effective, and people look for those signs very intently, but that's just not the first priority. The priority is first to test if it's safe, and then hope that there are some signs of activity. If a drug is safe and there are maybe some signs of activity, or there's hope for that, then it then will go on to a phase 2 study where you're really then looking for whether this drug is effective. And there's a lot of different ways to do that. You can compare it against standard treatment. Sometimes they just will give the drug to a larger group of patients and kind of have a benchmark. They'll say, we hope that 30% of the patients will respond, and whether that happens or not decides whether a drug then goes on to phase 3. And phase 3 is really often what's considered the benchmark to decide whether a drug is approved by the FDA. And that's comparing it against the standard treatments that are out there, or newer treatments that are out there, to see if that drug is as good or better than those treatments, to see if it can be adopted, widely. Sometimes drugs are adopted just based on phase 2 trials. That's true for some rare cancer types. So it doesn't always have to go to phase 3, because phase 3 is a big effort, obviously. Sometimes hundreds and thousands of patients have to be treated. But and for rare cancers, that can be very difficult. So sometimes, for example, in sarcoma, there was a recent phase 2 study that tested immunotherapy for approximately 120 patients or so. And the results were promising enough that I think that will likely lead to FDA approval for that indication without having to go to a phase 3, because sarcoma is a fairly rare disease. So these are kind of how they're divided into phase 1, 2, and 3. Phase 4 is kind of a follow-up situation where we look back at how patients do when they're actually treated in the real world, but that's less kind of critical for the development of the drugs.
Great, thank you. It's clear that it's a very complex process that takes a lot of planning, and I would love to ask you a little bit about the role that you have played in clinical trials and getting them off the ground and implemented.
Yeah, I think the reality is, because it's such a complex process, oncologists often will focus on one end of the process or the other. And my experience has been primarily in phase 1 clinical trials, and some in phase 2, which is to test out new drugs or new approaches for the first time, to understand the safety and hope for some signal. And because I run a research lab, that works really well with kind of my interest, because we come up with potentially new strategies and we want to test those in the clinic. And so the first test is phase 1. So going from the lab, it's most natural to kind of do phase 1 studies. And then you hand it off to other teams that are a little bit more expert in running the larger trials, the phase 2, 3 trials, if it needs to go on. So that's been my area. I've led phase 1 studies of immunotherapies for patients with solid tumors, including melanoma and sarcoma. And we've also led, specifically studies around T-cell therapy, or engineered T-cell therapy, called CAR T-cell therapy. So, and we'll be talking about that a little bit today. But phase 1 studies are challenging because, you have to do a lot of lead work to convince the FDA, first of all, that this drug might be safe. They won't just let you try a drug because you say it's safe. You have to kind of show them a lot of evidence that a drug might be safe, and then it might be effective. And then they'll give you the green light to then try that trial. And so that's a lot of lead work to get to that point. And then when you run the trial, there's a lot of requirements in terms of how you report, like, what information you have to collect from the patients to prove that this is safe. All the samples you have to collect from patients, the lab tests that you have to do, the visits you have to have with patients very closely to monitor if they're having side effects or not. So it's a pretty intensive period of time when patients are involved in phase 1 trials. Whereas in phase 2 and 3 trials, because drugs are generally safer or have already been proven to be safe, the emphasis is more on just, understanding if the drug works. And so there's usually not as much of a, meticulous look at how patients are doing from a side effect profile. So that's one of the challenges of phase 1.
Great. Well, thank you. Clearly, as a PI or somebody that's involved with implementing a clinical trial, you have really a deep belief or a really good hunch that there's something in there that could possibly work better than the standard of care. So I'm sure that's
Yeah.
really, really exciting and we're so grateful, for all of you that do this type of work.
Yeah.
You touched on,
Let me just add that I think, in fact, how hard it is to run these trials makes you really think as a BI, I better believe this has a good chance, because why would I do all of this and all these people that need to be involved in creating these trials and going through this process if I didn't think that these trials would have a chance? And so especially as you go on in your career, you really want to take shots on goal that are
yeah.
really going to make a difference for patients. And so, sometimes it can be a daunting road, but I think it also helps you make sure to check and think about what are the ideas that you want to bring to patients and make sure that they're your best ideas.
Well, that's really important to hear because I think that gives patients hope and gives them a lot of confidence in the process that, you all are working on things that really do have potential and there are better things to come on the horizon. It just takes time. So thank you for doing that. Let's
I,
talk about, oh, go
yeah,
ahead.
I was going to add one more thing, and kind of a nuance that maybe patients don't hear about so much is there's two kind of types of, or there's different ways trials can be sponsored. So in order to do these trials, it requires a lot of resources, financial resources being one of them. And so sometimes trials are run by companies because companies are often the ones developing drugs. And they're run at academic or non-academic centers, and people like me will run the trials on behalf of the company with some relationship with the company. Other times, there are trials that are actually started at institutions, at academic institutions, by academic investigators, and they're funded by organizations like MRA or other, philanthropy groups or NIH. And it is important to understand who is funding the research that is being done and where that, what is the driver of that research. And so, we have amazing success stories in both, company run trials and drug development, and as well as, investigator initiated trials. But I think it's a good thing for patients to be aware when they're looking at trials to understand.
Yep, that's a great reminder. Well, we've touched a little bit on CAR T cell therapy at the beginning, but I would like to dive deeper into that therapy and the trial that you are actually running and what problems that you seek to explore and identify solutions for. Can you
Sure.
tell us a little bit about it?
Yeah, why don't we pull up, I think there's a slide we have on just kind of a general overview of CAR T cell therapy that we can walk through.
Is
So,
that a better view for everyone? Just want to make sure.
that looks pretty good.
Okay, awesome. Thanks.
Um, so, most drugs are manufactured at a company and then they're just given to a patient, like a pill or an IV, and that's pretty straightforward. What we're working with here are cell therapies. So they're not your standard drugs because cells are living things, and these are administered to patients in a slightly different way, especially because in CAR T cell therapy, we're actually using cells from a patient's own body, and we're actually modifying them in the lab and then giving them back to the patient. And so it's kind of a complicated process, but the idea is that there are these cells in your body, a part of your immune system called T cells, and T cells have been shown to be really important and effective fighters against infection, but also against cancer. And so if the T cell has the ability to recognize the cancer, it can effectively kill a cancer cell, and it can kill many cancer cells. So people have been trying to harness this potential of T cells for a long time, and one of those ways is to take the T cells out of the body and give them the tools to recognize cancer. And one of these tools is called a CAR, stands for chimeric antigen receptor. How do you give a T cell a CAR? Well, you have to put in a gene into the cell, and that can be done in a variety of ways, sometimes with viruses. And you put that gene into the cell, and you can see that in the top right there. That's done in the laboratory. So first you get the blood from the patient, take it back to the lab, you get the T cells, the gene that you want to put in there. It's called the CAR gene, and the CAR is then makes its way as a protein onto the surface of the cell, on the surface where it can then recognize things on the outside. And so then you grow these CAR T cells to large numbers, millions of them, and then you put them in a bag, like other drugs, but now instead of a drug that's not living, you have actually living cells in that bag, your patient's own living cells, and they're then infused back into the patient. Often the patient has to get a little bit of chemotherapy beforehand to create kind of what we call space for the new cells to grow. And so then the CAR T cells go floating around the blood looking for the cancer cells. And because they have these CARs on the surface, they can see certain molecules. This is usually one specific molecule that they're trying to find that's expressed, that's on the surface of the cancer cell. So once they see that molecule, they can then attach and kill the cancer cell. And so that's the idea. And the cool thing about the CAR T cells is once they kill one cancer cell, they can move on and kill another cancer cell. And they can do this serially. And together with these millions of CAR T cells, they expand inside the patient to even larger numbers. And the idea is they can circulate around the body and kill any cancer anywhere in the body. And the idea is that they target just the cancer cells and not any. These proteins that they're targeting on the surface of the cancer cell is not on normal cells, so it could potentially be safe. And so that's the idea of CAR T cell therapy, and it's been, revolutionary for certain types of cancers, especially blood cancers, like leukemias and some lymphomas. And so there's been a lot of excitement around CAR T cell therapy and a lot of companies around CAR T cell therapy. But for cancers that are solid, things like melanoma, sarcoma, other tumors that are solid, pancreatic cancer, breast cancer, CAR T cells still haven't quite, had a huge success. We've seen glimmers of hope in the last year or two with a couple of types of cancer, solid cancer. And so there's a lot of progress being made on this front. And so this is one area that we've been working on in patients with melanoma to try to bring CAR T cells to melanoma, which haven't yet been successful in patients with melanoma.
Wow, thank you. It's really interesting. So it was the leukemia and lymphoma successes that kind of led you to believe that there might be some efficacy for the solid tumors, and that's why you are conducting the research?
Yeah, that and the fact that in melanoma, T cells are also effective, but instead of T cells that are engineered with CARs, they're actually T cells that are grown from inside of the tumor. This is called TIL therapy, and tumors are removed from patients with melanoma, and the T cells are grown up to large numbers outside of the patient and then infused back to the patient. But these are the patient's own natural T cells. They just happen to be in the tumor. But we thought, well, if the T cells are able to do this that are naturally there, if we give them the CAR, they may even work even better. So the idea is then to kind of build on this early success of TIL therapy in melanoma and use this as another alternative for patients where TIL therapy doesn't work. Because in TIL therapy, you're kind of relying on your T cells to naturally recognize the cancer. Not all patients have T cells that naturally find and kill cancer cells. And so CAR T cells allow us a way to artificially give the T cells a way to see the cancer.
That's great. So it sort of takes away a little bit of the subjectivity from patient to patient.
Exactly, that's the idea.
That's great. So if I'm not mistaken, this is a phase 1 trial, correct?
Yeah, so we're doing a trial that's been a really fantastic collaboration between UCLA, where I used to be, and City of Hope, and Stanford, where I am now. And we're using a CAR that was actually developed for brain cancers and has had some success in glioblastoma type of brain cancer. And so that was developed at City of Hope by one of my colleagues, Christine Brown. And then we learned that the target of that CAR, which is called IL-13 receptor alpha 2, it's a long name, but that target is actually also a target on patients with melanoma. So melanomas also can express this target about 1/5 of the time. So about every 5 melanoma patients has this target expressed. And so we thought, well, that's great. We already have a CAR and it's been tested in brain cancers. Let's try it in melanoma. The big difference is in brain cancers, the CAR T cells are just given directly into the brain. So they're not given into the blood. So we had to go to the FDA and ask them, well, can we try this? Could we try this to test if it's safe to give it in the blood for the first time? So that's why it's a phase 1 trial, because we're the first time to try these CAR T cells in the blood. And we've so far treated 7 patients. And so we're making progress, and now we have funding to try to actually make it a trial that's going to be open. One of the first trials for CAR T cells open at 3 different academic sites at the same time, across State of Hope, UCLA, and Stanford. So so I think it's a great opportunity for us to learn about CAR T cells and melanoma and make some progress on that front.
Yeah, that really brings to life the whole phased approach to trials. So thank you for explaining that. Are you targeting a specific type of melanoma?
No, so far what we can tell is the target is IL-13 receptor alpha 2, which is the target we're going after, is expressed on different types of melanoma. We've had patients with mucosal melanoma, uveal melanoma, and cutaneous skin melanoma that have had this target. So it's not exclusive to one, but it doesn't exclude any of the kind of rarer forms of melanoma either.
That's great. That's really great.
The good news is we actually also know that now this target is expressed on other types of cancers, not just melanoma as well. So there are other patients with solid tumors that may be eligible for this trial. Things like adrenocortical carcinoma, paraganglioma, pheochromocytoma. There's some other rare tumor types that express this target. So we're also doing outreach to patients of different types, those cancer types to see if they
yeah, that's fantastic. I know a lot of melanoma research has successfully led to treatments for other cancers, and hopefully this is one where, anybody that's part of the research now will impact others down the road. So that's really exciting. How is eligibility determined for patients that want to be part of this trial?
Yeah, so we just have, first of all, it has to be a patient that needs a therapy, so their cancer has to be progressing. We also have to do testing of their tumor to see if that target is actually expressed. It's only about 1 in 5 melanoma patients expresses the target. We wouldn't want to treat all melanoma patients because it would probably be not helpful for patients whose tumors don't have the target. So we do a quick test using, if you have tissue that's been biopsied in the past or you've had surgery on a tumor that's stored at some center, we ask for that tissue and we do a quick stain of that tissue to see if the target is there or not. And if it is, then the patient would be eligible. And then we have to check some other basic things to make sure this patient is healthy enough. We do have an age cutoff of 75. Other than that, there are kind of some standard things to make sure the patient's healthy enough to undergo the treatment.
Great, thank you. So other than what you just mentioned, there's no particular pre-screening. Is that right?
Other than the test of the tumor, that's the pre-screening.
Yeah. Okay, great. Thank you.
And the great thing is we have a pretty easy way to get the pre-screening done remotely. So it's a phone call with our study team, and then they'll get your information and request it from whatever center the tissue is at. And then within a couple of weeks, we usually have the results. And then we let your doctor, and then you can discuss with your doctor whether you're interested in pursuing the trial, and then we move forward from there.
That's great. And that marker usually shows up if it's there. It's not something that would give you a false negative or
no, occasionally
or a false positive.
Occasionally patients, will have a specimen that didn't have enough tumor for us to assess or something like that, but the vast majority of the time that's not an issue. The other thing I would say is it's also great to know if you would be eligible, even if you don't maybe need the trial right now, because you may need it in 6 months or a year or 2 years or down the road. And it doesn't hurt to test earlier and to just have that, as an option down the road, should the current treatment that you're on not stop working or stop being effective.
Absolutely. I think that's really important. Always have something in your back pocket. Exactly. That's great. Any other thoughts that you would like to share based on the trial that we didn't touch on?
Yeah. So, we have treated the last, I think, 5 or 6 patients and done really well in terms of without any side effects, and the CAR T cells expand in the patient. So we're really excited about the potential safety of it. And we've seen some glimmers of responses in a few of the patients, although we've been at a very low dose, because when you do these phase 1s, you have to start at a low dose. And now we're just starting to get to a higher dose. So we're really excited about the potential benefit that it may have for patients now that we're getting to the higher dose.
That's great. It's wonderful to hear that success. I think we received a few questions from people that registered. Renee, can you please share the first?
Yes, certainly we do. We have a couple, and then I actually have a question of my own when, we get through some other questions. So this question is kind of alluding to the eligibility requirements for the trial and just trial eligibility in general, because, stating advanced melanoma, this person wants to know, is there a significant difference when discussing advanced versus stage 4 melanoma in trial eligibility, or is that just a matter of semantics?
Generally, advanced it definitely means stage 4, but sometimes when we say advanced, it can also include some patients with stage 3 disease. Usually in the actual fine print of the protocol, it'll map out exactly what advanced means, but sometimes in the patient section, just to avoid too much jargon, they'll just say advanced because that kind of captures, it's a little bit more vague, so it kind of captures more possibilities. But in the fine print for our study, it's, for example, at stage 4, as well as some patients with very advanced stage 3 disease.
Okay, thank you. Another
Great.
question we received ahead of time touches on something you mentioned earlier when you mentioned that the rare subtypes would be eligible for this type of therapy. But what if an individual with one of these rare melanoma types previously received TIL and their tumors progressed?
Yeah, those are exactly the patients that we're looking for this, because
Great.
we think that potentially this therapy might be effective in patients where TIL therapy was not, or where, the other immunotherapies were not effective, like Keytruda or Opdivo or any of those. So certainly this is exactly what we're looking to test this on.
That's great. And that's just for clarification, because technically cell therapy is considered as a type of immunotherapy. So when you all say progressed on immunotherapy, that can include another type of cell therapy.
That's right. It can progress really on any therapy, would be eligible for our study, whether it's immunotherapy or not. But generally, melanoma patients will have received some immunotherapy before they get to us, since that's the standard treatment. And so, yeah, any immunotherapy or TIL therapy prior TIL therapy would be fine if we progress on those therapies. Generally speaking, I think now patients will probably want to try TIL therapy before they try our trial, just because it's now FDA approved. So it's a standard treatment. Sometimes patients can't have TIL therapy because the TIL doesn't grow, or they don't have a good tumor to have the TIL removed. So there are some cases where a patient may not be able to get TIL therapy. In that case, they may be eligible for a trial.
Great. And then I just want to encourage everyone, if you want to drop a question in the chat, if you want to be anonymous, you could just send it to me directly, or you could raise your hand. But another question, you mentioned that 1 in 5 of melanoma patients have the specific target that you're looking for.
Uh-huh.
What is there a difference between like looking for this target and looking for something like a BRAF mutation? Like, are they different?
Yeah, it's a little bit different. BRAF mutation is generally thought to be, when it happens in all the cells, and it's actually a driver of the cancer. It's what causes the cancer. What we're looking for is more of a flag on the cancer surface. The flag on the surface itself doesn't do anything. It's just kind of a sign that the cancer is there. So in that sense, we're just targeting something on the surface, rather than a BRAF is a molecule inside the cell that actually makes the cancer go. And so using a drug to stop the BRAF is effective in melanoma, but a drug to stop this target wouldn't be effective. Well, we're just using it as a way to find the cell and kill the whole cell.
Awesome, thank you. That's all I have, but I want to see. Oh, we just got a question in the chat. Do you have to be on the West Coast for this trial?
Well, you don't have to be to enroll, but to get the treatment, yes. Right now, the trial is only open treating patients at UCLA. Hopefully, very soon, we'll be treating patients at Stanford and City of Hope. But we do have funds for travel and to support patients lodging while they're getting well, they're getting the treatment, so to defray the costs, because we know that's a big issue. And that's an important thing for patients to ask about when they're thinking about trials, is what are the resources that they may have for patients. In our case, we do have resources to, capture patients from all over. So if that's something that, you're considering, we can do the pre-screening process remotely, so you wouldn't have to come, but once you would want to actually enroll, you'd have to come out.
That's really generous and good information to have. I think we got another question. What makes a cancer a hard tumor?
Yeah.
You
Yeah.
mentioned melanoma versus pancreatic and other solid tumors.
Yeah. So yeah, hard or solid is the term we often use, but it's a way that we group cancers generally, and basically it's a group of cancers that arise in the blood from cells in the blood. So that's what makes them kind of quote unquote liquid, because they're in the blood and they float around. Whereas solid tumors are tumors that kind of form a mass. They form a lump or a nodule or a tumor. So any of these now doesn't mean they're all actually hard. Most of these are actually, relatively soft. And some tumors are harder than others, but it's just a way for us to categorize the blood cancers, the liquid tumors, versus the more mass-like tumors. So we call them solid. Now, it's important from an immune system perspective for a T cell, it's harder to get inside of a solid mass and kill all those cells, because when the tumor forms and makes a hard mass, it creates this ecosystem. It's an environment that's actually kind of hostile to the immune system. So that's one of the reasons CAR T cells may have a harder time killing solid tumors than they do killing leukemias, which are these cells that are floating around the blood. They don't have this whole ecosystem protecting them. So that's one of the challenges, and we're coming up with strategies to tackle that. So, but that's an important distinction.
Thank you. Any other questions? Looks like there's one more.
Yeah.
Just reading it.
I'm just going to read it out loud for the sake of the
Okay.
recording. So the question is, in regards to how Dr. Kalbasi stated that CAR T cells bind to and kill the cancer, is it known if they do this proactively, or can this not be determined? In other words, is it yet known how long the effectiveness lasts? Does this generate ongoing T cell production?
That's a really wonderful question. One of the, reasons we're so excited about cell therapy or these living drugs, is that the idea that T cells, just like when you encounter an infection, T cells protect you for the rest of your life. They circulate and expand, and then there's this population of T cells that becomes memory cells, and so they stay around. So the next time you see that infection, they deal with it. The idea with CAR T cells is that this could happen as well, that you could get the CAR T cells, proactively kill the tumor, and then proactively stay around the body for the rest of your life and serve as a line of protection against any cancers that have that target on them. So certainly that's the holy grail. Now, whether that's happening with the current iteration of CAR T cells is debatable. Some patients with leukemia, certainly we've seen that. With solid tumors, we're not quite there yet, but that's what we're working towards.
Great, thank you. One more question. Who do we contact for the remote pre-screening? And do you have any patients on this trial that have the BRAF-K mutation specifically? I've read that the K is far less common than the V.
So that's a great question. First of all, regarding the contact, I can share with MRA the email contact for the study to inquire about pre-screening, and they can disseminate it somehow, either to the people who joined the chat, or I'll let them figure that out. And then in terms of the patients on the trial that have had BRAF mutations, we've had patients with BRAF mutations. I don't think we've had any with the K mutation, but it's correct. The V is the more common one.
Great, thank you.
Let me actually, I can share another email specifically in the chat for who to contact for inquiring about the trial.
Awesome. Thank you.
Yeah, that's really great. We so appreciate your time and your expertise and your insight and your willingness to solve some of these really big problems for melanoma patients and beyond. Thank you so much for your time, and we thank everybody for joining. And look ahead, we'll be holding more webinars starting January, so we will look forward to seeing all of you there. This will be posted on our website, though, in case you would like to refer back.
Thank you so much for your time and everyone for their time and great questions.
Thank you,
All
Dr.
right.
Kalbasi.
Thanks, everyone. Happy holidays. Bye-bye.
Bye.